Study topic number 4 goes along with Unit 4 from the AP curriculum. If you remember, I kind of split Unit 4 into different sections, and we talked about cell communication first, and we did things like the signal transduction pathways. I think that was the first time you did the Plato videos, and then we eventually talked about the cell cycle with mitosis and meiosis. In this video, we're going to look at all of that together as one unit. So this first slide looks at just an overview and how those two processes about the cell signal transduction pathway and the cell cycle all kind of work together. So when we're talking about cell-to-cell communication, cells in a multicellular organism or even unicellular organisms need to communicate with each other because all of the processes that are going on inside of the cell are controlled and they are regulated. So photosynthesis is regulated, cellular respiration is regulated, gene expression is regulated, mitosis is regulated. And how do cells know when to either do these processes or stop these processes? That's where cell communication comes in. So cells can communicate with themselves, they can communicate with each other if they're close to each other, or if they are far away from each other. And the chart on the left-hand side kind of reviews that for you. One of the things that is a process that is regulated by cell communication is the cell cycle, right? So looking at when a cell goes through the cell cycle, how does it know when to continue through the cell cycle, or how does it know when to pause? That's where cell communication comes in. So as you've seen in the last few videos, here's a list of some questions that could appear on the exam. Again, you're not necessarily looking for details of the phases of mitosis and meiosis, but kind of the big picture of everything. So take a minute, pause here, look at these questions, and we will be hitting most of them in this video. As you also saw in previous videos, there is a list of vocabulary that is covered in Unit 4. You again probably want to stop here, pause here, and then reach out with whatever questions that you may have. Quick refresher of a signal transduction pathway all starts with how cells communicate. We talked about autocrine signaling. That's the first picture up here, where a signal molecule is created by the cell and the target cell is itself. We see this a lot in cancer cells where they signal themselves to go through the cell cycle and to go through mitosis. Different types of direct contact we can call gap junctions, where they're actually attached to each other. And one cell can send a signal through that gap junction to the other target cell and talk to it. Paracrine signaling is when cells are close to each other but not necessarily touching. They call that short distance. And then long distance, think of the endocrine system where hormones and signaling molecules are sent from one part of the body, such as the thyroid, and those signal molecules travel all through the bloodstream long distance to the different target cells. So we know that those processes involve the signals, which we also call the ligand, and the receptor proteins. Those proteins can either be in the membrane or they can be inside the cytoplasm. The ones that are in the membrane, we looked at G proteins, we looked at the ligand-gated ion channels, and we also looked at the receptor tyrosine kinases. Think of the Plato model that you did and what your specific protein was. There are also intracellular receptors that are proteins. For example, in this figure, they're showing you that the receptor is inside the cytoplasm and the signal is entering through the membrane and being accepted at that receptor. We also talked about the signal transduction pathway, where those relay proteins, those kinases, turn each other on and amplify that signal, making a big cascade. Different things that can be our response, maybe those end proteins at the end of the cascade are transcription factors that will go and turn gene expression on. Possibly those end proteins at the end of a cascade could be enzymes that get turned on. We looked at all kinds of different responses that can happen as a result of the signal transduction pathway. So here's a question from the 2013 exam. This would be one of the shorter type questions. And really, this is one that's just straightforward. You don't really have to figure anything out. You're just looking at what does this figure represent, and then explain the role of each step. So take a second, plan your response, write a little response down, and then check your answer on this next page. So looking at the answers, this would be an excellent question that I know you all could answer, especially after we did that stop motion video, right? You all know that this first step would be the reception, where we're receiving the signal. That's going to trigger the cascade, or what we call transduction, and that signal would be amplified. And eventually the response in step 3 is the turning on or the expression of genes. So this would be a fantastic question if you would get this on the 18th. Extra credit or extra content for you to take a look at are just some extra questions. For example, number 1, is this protein signaling, is it protein or is it a steroid? And thinking back to, we did the case study with the My Dog is Broken, if you remember that case study. That case study actually looked at the ligand entering through the cell membrane and being received inside the cytoplasm. If that is the case, that would be a steroid signal, right? Steroids are lipids and are able to pass right through the membrane. If this was a protein signal, then our ligand, our signal, would be received in the membrane. So looking at our figure, where is this being received? Is it being received in the cytoplasm, or is it being received in the membrane? This case, it's the membrane, so most likely this would be a protein ligand or a protein signal. Number 2, what are other possible names for the molecules in step 2? We looked at these being called kinases. We looked at these being second messengers like cyclic AMP or CAMP. You could have mentioned any of those. And then number 3, what type of signaling is most likely occurring here? Is this going to be autocrine? Is this going to be endocrine? And then how do you know? Because we're looking at this coming from the extracellular fluid, this is most likely not autocrine. If it was autocrine, we would see the signal coming out of the cell and then being received by the cell. But because this looks like it's coming from somewhere else, we can assume that it's endocrine, or we could even say paracrine, but that wasn't really an option here. But the fact that it's not coming from the cell itself, it's coming from somewhere else, we would say that it's not autocrine. And then this is a specific type of response, right? They're looking at a gene being turned on, but there could be other things that are going on. Here are some examples here. This could start cell division. We could see protein being produced, enzymes being produced, all kinds of different things being made from that gene expression. Okay, so that's your first little question here that goes with signaling and those types of pathways. The second section of this looks at the cell cycle, mitosis, and then cancer, right? So we know that cells receive signals for them to proceed through the cell cycle. If they get too large, they will go through the cell cycle. If there's an injury to the body, mitosis will happen. If there's growth needed, the cell cycle will proceed as well, okay? Now, in the curriculum, they tell you that you do not have to memorize the phases. So there will not be a question on there that says, what happens in anaphase? But they could possibly ask you, like if you look over here on the side, what errors could occur? So what could happen if the DNA synthesis, so the S phase, is not happening correctly? Or if we're in mitosis and the chromosomes don't align in metaphase, what could happen? So those are the types of questions that you might see. And coming up is a question from a fairly old one from 2015. So for both mitosis and meiosis, these are both types of cell division that have daughter cells. For part A, describe 2 events that are common and how they ensure that the resulting daughter cells inherit the appropriate number of chromosomes. And then part B, the number of chromosomes or the genetic composition of those daughter cells is different. Describe 2 features that leads to these differences. So this is a nice little section where you could brainstorm and make a little table. But remember that on the AP exam that you have to write your answers in sentence form in order for them to be scored properly. So pause the video here, see if you can come up with similarities and differences here, and then think about how does mitosis differ from meiosis as far as chromosome numbers, and then where does that come from? Again, take a look at the answer on the next couple slides, and then we will talk a little bit about it. So we know that mitosis starts with a diploid number of chromosomes. Through one cell division, one PMAT, so think one prophase, metaphase, anaphase, telophase, and then cytokinesis, we end up with 2 daughter cells that are still diploid. So if we're talking humans, we would start with 46 chromosomes, and each daughter cell would end up with 46 chromosomes. In meiosis, we start with the diploid number, but remember, this goes through 2 meioses. We call them meiosis 1 and meiosis 2. And the key difference is that in meiosis 1, those homologous chromosomes find each other. So we talked about, let's do like red for mom and blue for dad. So every chromosome pair, for example, if this is chromosome number 1, the mommy chromosome finds the daddy chromosome and they pair up. We can also see some crossing over where the chromosomes are touching. We call that a synapse. And that is very, very different from what happens in mitosis. Meiosis 1 is completely different. So we have the homologous chromosomes pairing up. Each of these chromosomes pulls apart. So the blue one separates from the red one. And in each of the daughter cells, we now have half the number of chromosomes. So we say that in meiosis 1, this is our reduction division. We're going from diploid to haploid. Meiosis 2 is almost identical to the processes that occur in mitosis. We don't have homologous chromosomes pairing up. They line up by themselves at metaphase, and then each daughter cell ends up with a haploid number. So if this was humans, again, we would start with 46 chromosomes in our original cell, and then each daughter cell would end up with 23. So looking back at the rubric, we can see what they were trying to get you to think about. So what was different about mitosis versus meiosis for part B? Here's some extra practice if you'd like to work through these, just so that you can refresh your memory about the phases of mitosis. And then as you saw in all of the other videos and all of the other PowerPoints, this is directly from the AP curriculum. So this might be a nice little checklist for you to go through and say, hey, yes, I know this, but I really need to study this. And that would be a nice point to help you study for your upcoming exam. Thanks, y'all.